I invite Dr. Neeraj Sooj and Dr. P.K. Patil to coordinate the travel award session.
So, good evening, everybody. Now we will start with the evaluation of travel grant awards. So for that, I am requesting Dr. Subdeep Ghosh, Chair. Ghosh. लगाओ फोन लगाओ घोष को फोन दो हाँ सर आई प्लीज डॉक्टर एनपी साहू sir.
Very good evening. It is really great privilege to welcome our chairs. It's my pleasure to introduce Dr. Narottam Prasad Sahuji, who is the Acting Director of ICR Center Institute of Fisheries Education, Mumbai. He is a leading nutritionist in the fisheries sector with experience of over 30 years, and he is known for his pioneer work in nutraceuticals, medicated feeds, and species-specific aquafeeds. He has been a recipient of several prestigious honors, including the Dr. M.S. Swaminathan Award for the Best Fisheries Scientist 2006, Achievement Award by Compound Livestock Feed Manufacturers Association 2014, and multiple institutional recognitions by ICR Center Institute of Fisheries Education. He is a fellow of both National Academy of Veterinary Sciences 2015 and Maharashtra Academy of Sciences. His scientific contributions include more than 400 publications, 2 patents, and several commercial aquafeed technologies. Sir, we are really grateful to you for kindly agreeing to chair this session. Thank you, sir.
I also take this opportunity to introduce our the second chair, Dr. Subdeep Ghosh, who is currently serving as the Assistant Director General, Marine Fisheries at the Indian Council of Agriculture Research, New Delhi. He is pioneer research on climate change impacts, carbon footprinting of fishing harbors, sustainable marine cage farming, has shaped the national strategy for fisheries management. A recipient of Pillai Aquaculture Award and Shri JVH Dikshitulu National Award, Dr. Ghosh has also led several national and internationally important projects focusing marine resources, sustainability, and aquaculture innovations. His expertise and leadership continue to influence the policy, research, and practice in India's blue economy. Sitting in the SMD, he is steering all the major fisheries and aquaculture activities in the country. I request both the chairs to coordinate this travel grant presentations and help the session. Thank you.
Hello. Thank you. Yeah, it's okay. So friends, we'll start with this travel award session. In this, we have around 7 presentations. Already we are running short by 1 hour. So I would request the presenters to be very crisp as possible, so that maybe we can finish by around 8 o'clock. So as I mentioned, we have 7 presentations. Shall we start the first one, or you want to give some opening remarks? So we'll call the first presenter, Bhupendra Singh, who will be presenting on development of autogenous vaccine against warm water lactococcosis disease caused by bacterium Lactococcus garvi in farmed rainbow trout in the Indian Himalayan region. So I would request Dr. Bhupendra Singh. Yeah.
So in this presentation, I request this total presentation you keep for 7 minutes and 3 minutes for discussion. So in 10 minutes, you should be able to.
Good evening to all delegates. Myself, Bhupendra Singh, currently working as a junior research fellow at ICR CISFR and a PhD scholar at Kumaon University in Nainital. Today I am here to present my manuscript titled as Development of Autogenous Vaccine Against Warm Water Lactococcosis, which is caused by the bacterium Lactococcus garvi in farm rainbow trout of northern Himalayan region of India, an alternative to antibiotics. The disease Lactococcosis has a prevalence in rainbow trout, and it has been reported from 17 countries all over the world. Among those 17 countries, India has reported this disease in 2018 from rainbow trout. These are the gross clinical signs for the leptococcus diseases, which has exophthalmia, acute septic hemorrhagic conditions in buccal cavity and the corneal cavity of the fish. In this study, the sampling side of our study was from Himachal Pradesh and Uttarakhand. Here, we have taken our isolate from Himachal Pradesh, where we found the isolate from an infected rainbow trout farm, and we got the isolate, which we are using as an autogenous vaccine, and the strain has been used for this study. The economic loss which has been caused by this disease is very high. As we can see here, the production rate of the rainbow trout is almost 4,800 metric tons annually, which was reported by the PMSY, and the value of this production is around 240 crores, which is equivalent to $27 million. It has a very direct mortality, which causes due to the high temperature that cause in Trans-Himalayan region at around 20 degree, which cause around 85% of the total mortality in the farm, which has also been reported in the Himachal Pradesh. Here, the normal estimated production loss due to this leptococcus is around 10 to 20% of all. Here, the 900 metric ton of annual production rate has been dropped down because of this disease, and which has an economic loss of $2.8 million. This is due to the global temperature rise and intensification in trout farming because of the higher production rate. The current mitigation strategy against leptococcus is being used as an antibiotic used in feed, which has been very higher concentration as compared to the other antibiotics. And currently, there is no such commercial vaccine available for the leptococcus in India for rainbow trout. The objective of this study is that we are trying to develop an inactivated immersion vaccine to assess the vaccine efficiency, biosafety, and the vaccine efficacy. The methodology of this study has been done here, where we have taken the 400 rainbow trout fries as a samples to which we will go to vaccinate. We will try to vaccinate them. And the selection of bacterial strain here, we are using the Kg positive, which is a non-capsular strain of Lactobacillus garvi, which has been isolated from Himachal Pradesh trout farm. The vaccine is prepared with the using of formalin-killed method. Here we have used 0.75% of the formalin and have incubated it at 27 degree for 3 hours in a shaker incubator. After that, we had done the biosafety and the efficacy and the data analysis. I will directly go to the results section of my study. In results section, we have checked the biosafety of our vaccine in vitro condition in Epithelioma papillosum cyprini cell lines. Here we have checked the biosafety of this vaccine in 2 different type of dilution, in 10-fold decreasing dilution and 50-fold, 2-fold increasing dilution. Here you can see we have checked the dilution in 72 hours post-infection and 96 hours post-infection. Here there was no cytopenic effect was found as compared to non-vaccinated fish, and the cell maintained the normal morphology and confluence, and inactivated vaccine did not show any effect in cell viability. We also have done the MTT assay, which has no significant changes and no cytotoxicity. Rizazurin assay was also been done and has shown no difference in cell viability, and the cell viability was always more than 90% in all the groups, in vaccinated group and in non-vaccinated groups. In vivo condition, we have checked the biosafety of our vaccine in rainbow trout tissues, which has directly contact with the immersion vaccine, like gill, muscle, eye, and also we have checked in the immunogenic organs like kidney, spleen, and also the hepatic pancreatic cells in its liver. And there was no histopathological alteration was found. This is the general stress we have recorded, stress behavior we have recorded. There was no such stress behavior was found. Everything was fine, and there was 0 histopathological scoring in all the tissues. Now we go to the vaccine efficacy. We have checked the vaccine efficacy for almost 120 days post-challenge. Here you can see we have got the RPS at around 83.78%, and the cumulative mortality was noted as 3.84 for vaccinated group, which has been challenged with the bacteria, and the non-vaccinated group showed 23.731 cumulative mortality. The humoral immune response was checked in the blood serum parameters. In lysozyme activity was checked against the Micrococcus luteus, and in which the vaccinated group has showed the higher significant changes as compared to non-vaccinated group on 3 and 5 days post-challenge. In bactericidal activity, after giving the challenge, the vaccinated group has shown, we have checked the bactericidal activity in plate count method. We can see that the vaccinated group has shown the lesser growth of Lactobacillus garvi bacteria afterwards in the bactericidal activity. And it has been prominent in 3rd, 5th, and 7 days post-challenge. We also checked the serum antibody titer, which was higher in vaccinated group, which was peaked at 5th day post-challenge. We also checked the adaptive immunity using the immune gene expression in 2 major immunogenic organs, first in spleen and another in anterior kidney, which is also known as head kidney. The significant upregulation of pro-inflammatory cytokines like IL-6, 8, 21, and tumor necrosis factor alpha-1 was seen very high as compared to non-vaccinated group, but in adaptive immunity markers like interferon gamma and immunoglobulin D and M showed very higher gene expression as compared to non-vaccinated group. In the case of anterior kidney, the early upregulation of in 3-day post-challenge was seen in pro-inflammatory genes like IL-6 and tumor necrosis factor alpha-1. The adaptive immunity markers IgM secretary showed the very spiked upregulation as compared to non-vaccinated group in anterior kidney. The conclusion of my study is that we have developed a safe and effective immersion vaccine, and there was no biosafety concern found in this study. We have found that it causes and it elevates the cellular and humoral immune response, and it is a sustainable solution for trout farming in India, in the northern region, and it is much more feasible to develop autogenous vaccine, as it is more, location specific, and it is much more easier and much more faster to make. I would like to acknowledge DBT that they have funded this study, and ICR-CFR to providing the lab facilities, DAA12 and Fish Health Section and AFS for providing the student travel grant, and Mr. Mukesh Bhatt for providing the field and laboratory assistance. At last, I would like to thank you for listening to me.
Thank you for the excellent presentation. Maybe 1 or 2 very quick questions.
And congratulations for such nice work. Only 2 things I wanted to ask you. Normally, the behavioral changes which are observed as a safety parameters, they are done within the 48 hours, like that. Because definitely if any adverse behavioral change is coming, that comes within 48 hours. And then when you want to prove that this is a biosafety, then what you do is behavioral changes within 48 hours and recovery period like 7 hours, 7 days. But your study is saying about 7 days only. So have you studied before that, which is not presented here?
No, ma'am. We actually did the biosafety on the 0th day when we gave the initial dose, and afterward on this booster day, we have taken the sample for the biosafety. That are the tissue.
No, not the tissues. I'm talking about the behavioral. The chart.
Behavior was that chart was for efficacy. Oh, sorry, that table.
Biosafety table.
Yes, sir. It was noted down at the day it started when we started giving the immersion vaccine.
Yeah. Okay. So any behavioral changes were there at 24 hours, 48 hours?
We actually give feed 2 times or 3 times a day. Afterwards, we gave the immersion. So in that time period, we try to note it down what are the behavioral changes, like it's coming on the surface of the water for gapsing or anything else, and any sort of melanosis.
The pattern.
So we have done in almost every day. Initially, it was done within 6 hours. because the immersion was given in the morning and we checked within 2-3 hours for checking and try to see is there any changes or any mortality is going on because of the vaccine.
Yeah, yeah. Okay, because if that would have been included, then you can say that this was the changes and recovery was like after 7 days there was nothing. That would have been more logical, but it's wonderful otherwise. And second thing is, do you have done any toxicity study or the safety study with higher dosage, like 2x or 5x, 10x like that?
No, ma'am, not right now. We haven't done that, but we are trying to check the LD50 dose also. You are asking for that, but we haven't done that part.
Okay, okay, thank you.
Thank you.
If there are no more questions, thank you, Dr. Singh, for the excellent presentation and the excellent work.
Second presenter will be Naritoyo Ishiba's identification and genomic investigation of a novel fish pox virus in hatchery red sea bream.
Thank you for your kind introduction. Good evening. I'm Naritoyo Ishibashi from Japan, and I'm master course student at Okayama University of Science. I'm deeply honored to have been selected for the Student Travel Award, and I'd like to express my sincere gratitude to the organizer for giving me this valuable opportunity. So now, I will start my presentation. To begin, let me introduce red sea bream. This is a saltwater fish, and it is popular in Japan. As you can see, red sea bream is the second most produced fish in Japan. So let me, so now, let's get down to my main topic. In 2020, a case of unexplained death was confirmed at red sea bream hatchery in Ehime Prefecture. At that time, about 90% of the 350,000 juveniles died within a few days. The diseased fish express skin darkening and sleep-like symptoms. This is a serious problem for the farmers because this disease has repeatedly occurred and sometimes cause most mortalities. Therefore, as the first aim of this study, we decided to investigating the causative agent. So initially, we carried out a comprehensive genetic analysis of the diseased fish using RNA sequence. Total RNA was extracted from 4 diseased fish, and cDNA library were prepared and next generation sequencing were performed. The obtained data were analyzed as follows. First, the obtained short reads were mapped to the red sea bream genome, and the remaining unmapped reads were assembled. We conduct the homology search against public sequence gene database. So here is the result of the comprehensive genetic analysis. A total of about 3,000 contigs were assembled, and most of them matched bacterial sequences. Of these contigs, 56 contigs showed similarity to salmonella pox virus, but the amino acid identity was relatively low. After that, based on these findings, we carried out a preliminary PCR test for this unknown virus, between 2020 and 2023. As a result of PCR test, 16 positive reactions were confirmed. Therefore, the causative agent of this disease is most likely the novel pox virus. From now on, I will call this virus Japanese Silvery Pox virus, JSPV. In this slide, I'd like to introduce the non-fish pox viruses. So far, 3 pox viruses have been reported in fish, Atlantic salmon, and carp, and ayu. All of them are double-stranded DNA viruses and mainly target geese. Fish pox virus isolation has not been established, so their definitive diagnosis is performed by PCR. So next, we focused on investigating of the genome of JSPV. Here is how to conduct the genomic investigation of JSPV. First, to obtain genomic information of this virus, DNA-seq was performed on 7 samples because JSPV is DNA virus. As in previous RNA sequence analysis, host-derived leads were removed and the remaining leads were assembled to construct the draft genome of JSPV. After that, gene annotation was performed. In addition, to clarify the taxonomic position of JSPV, phylogenetic analysis and comparative genomic analysis were conducted. Here is the result of whole genome construction. From sample 5, JSPV draft genome was assembled. The figure below shows genome map of JSPV. It has inverted terminal repeat at both of ends, and it's unique to pox viruses. Also, 338 open reading frames were predicted, and it are located between SGPV and CV. The chart on the left shows phylogenetic tree based on DNA pox polymerase gene. As you can see, JSPV formed a cluster with other fish pox viruses, and at that time, JSPV showed 62% amino acid identity with SGPV and 44% with CEB. On the right, you can see a comparison of the whole genome sequences of fish pox viruses. Comparative genomic analysis revealed that the genome synteny was highly conserved in the central region of fish pox viruses. In particular, JSPV and SGPV exhibit strong conservation. But in contrast, terminal region lacked shared homologous. Furthermore, DNA-seq analysis suggested that the presence of multiple viral genotype. Therefore, we performed genotyping of the virus. Consensus primer were designed based on the viral core cysteine protease gene, I7L, and PCR were conducted on 24 samples collected up to 2024. The PCR product were sequenced using Sanger sequencing, and the obtained sequences were used for phylogenetic analysis. Here are the results of the genotypic investigation. The phylogenetic tree were constructed based on the partial I7L gene. JSPVs were broadly classified into 2 genogroups, genogroup 1 and genogroup 2. Besides, genogroup 1 were subdivided into genogroup 1A and group 1B. The amino acid identity between genogroups 1 and genogroup 2 was 81 to 83%. Finally, we developed a multiplex qPCR assay corresponding to the different genogroups of JSPB and used it to measure the viral load. As you can see, the figure in below, primers and probe were designed for each genogroups. To quantity JSPB, we prepare plasmid DNA containing the target amplicons and then applied the assay on 24 samples. On the left, you can see the standard curve generated by multiplex qPCR assay. Both genogroups showed high quantitative performance. On the right, you can see the viral copy number from the 24 samples. The viral load ranged from almost 10 to the 3 to the 10 copies. So I'd like to close my presentation. Thank you for your kind attention.
Any question is there? Okay, one question will be there.
Thank you very much for very interesting talk. My question is, did you check the histopathology of the diseased fish?
Okay. During the mass mortality event in 2020, I was not involved in this research, so I don't know whether pathological examination were conducted. In addition, we have not yet obtained good quality samples, so the pathological characterization is unclear.
Okay, thank you very much. If there is no more questions, thank you very much. We will call to the next.
Yeah, so thank you very much. Nice presentation, nice work. Our third presenter is Dr. Kazuma Yoshimura. He will be presenting on the first report of Red Sea bream, iridovirus genotype 1 and 2 recombinant strain in Japan.
Can I? Okay. Thank you for introduction. I'm Kazuma Yoshimura, a doctoral course of student of Tamasat in Japan. Today's title is first report of Red Sea bream iridovirus RSIV, a genotype 1 and genotype 2 recombinant strain in Japan. So first, the megalocyte virus genus belong to the Iridoviridae family as a cause serious infection disease in fish. As shown in the left figure, this disease have been reported worldwide. Outbreaks occurred particularly frequently in Asia. So this genus, megalocyte virus, is classified into 3 clades: infectious spleen kidney necrosis virus, ISKNV, and turbot red body iridovirus, TRBIV, and Red Sea bream iridovirus, RSIV. So RSIV causes disease in the over 30 species of the aquaculture fish. Since the first report occurrence in Japan in 1919, the disease has been mainly observed in Eastern Asia. RSIB classified into genotype 1 and genotype 2 based on the DNA sequence of the major capsid protein gene and ATP as a gene. So genotype 1 had been primarily reported in China and South Korea. So almost also reported was in Japan approximately 30 years ago. So genotype 2, RSIV genotype 2 is widely distributed in Asia, have been reported in China, South Korea, and Japan, surrounding countries, and also recently it also be identified in India. So except for the Himalayan strain, this is the first detected in the RSIV. Genotype 2 was found in Japan. So previous research suggests genotype 2 persists around the aquaculture farm in Japan. So in 2023 and 2024, we obtained three RSIV positive samples during RSIV screening test, healthy fish at 5 aquaculture farms in Oita and Ehime prefectures in the western part of Japan. Among these RSIV positive samples, one sample have found to be PCR positive but negative in the fluorescence antibody test using M10 antibodies. Therefore, in this study, we suggest subject to RSIV positive sample to whole genome analysis and perform the phylogenetic analysis of RSIV. So in this study, I performed the whole genome sequence in case of genotype 1, Oita 2024 and Ehime 2024. RSIV genomic DNA was amplified by PCR using primer that spanned RSIV genome to the 12 fractions. So these fragments are sequenced by nanopore glitchon. In case of genotype 2 Oita 2024, whole DNA, total DNA was extracted from spleen and then sequenced by Illumina MiSeq. So here is a result. Here is a whole genome of 3 strain RSIV. So all strains have approximately 112 kilobase pairs, and GC contains about 53%. So the next, DNA sequence of MCP and ATP as gene, SNP1-2 region of the 3 strains were aligned with those of previous strains. 7 strain for genotype 1, 7 strain genotype 2, 3 strain of the ISKNV for the outgroup. Phylogenetic tree was generated by MEGA12. So this is a result. Here is a molecular phylogenetic tree based on DNA sequence of major capsid protein gene. Genotype 1 Oita 2024 was classified into genotype 1 clade. So while the other 2 strain was classified into genotype 2. The next, the phylogenetic tree of based on DNA sequence of ATP as a gene. So ATP as a gene, all 3 strains were classified into genotype 2. So the last, here is the molecular phylogenetic tree based on DNA sequence of SNP1 and 2 regions. Genotype 1 Oita 2024 is classified into the genotype 1, while the other 2 strains were classified into genotype 2. So in case of genotype 1 Oita 2024, the strain, the result of phylogenetic analysis revealed that this strain has a distinct genotype regions. Therefore, I calculate average nucleotide identity for each strain using whole genome sequence. And generate heat map is here. In ANI analysis, RSIV strain was classified into 3 grades: genotype 1, genotype 2, and intermediate type. So the ANI value of the genotype 1 and genotype 2 was 91%. Intermediate type and genotype 1 or genotype 2 was 94%. So in this analysis, genotype 1 OITA 2024 here is classified into the intermediate type with C. bacillus virus isolated from China and Korea, while the other 2 strains were classified as genotype 2. The next, for checking the homology among genotypes in detail, the whole genome was aligned using DIGAlign software with T-BLAST-X. The genotype 1 OITA 2024 genome contains regions that homologous to the type 1 red color one and type 2 blue color one. So, as well as one region, white color arrow region have a low homology to both genotype 1 and genotype 2. In case of genotype 2, OITA 2024 and EHME 2024, between these 2 strains, they show high homology. Also, they show high homology with genotype 2. The last, genotype 1 OITA 2024 is classified into the intermediate type. So I performed BLAST-N search on all predicted ORF to determine whether they show the higher homologous to the genotype 1 or 2. As a result, signal transduction cell cycle related ORF showed homology with genotype 2. The DNA replication transcription related ORF showed high homology to genotype 1. So in conclusion, different genotype of RSIV were detected in 2024 in Japan. The whole genome of genotype 1 OITA 2024 has region homology to genotype 1 and genotype 2. This is the first detection of this type in Japan. Thank you for your kind attention.
Thank you so much for the excellent work and the excellent presentation.
Thank you very much.
Maybe a couple of very, very quick questions, if you have any.
Yeah, thank you for that presentation. So as you may be aware that RSIV is now grouped in one species under WOA, that is infection with megalocytovirus Grus1. And this group includes RSIV, ISKNV, and DRBIV. So these 3 genogroups of megalocytovirus are almost similar to each other. So how sure are you that what you are dealing with is really RSIV and not ISKNB or DRBIV?
Thank you for your questions. First, I conduct a diagnosis test for the RSIV major capsule protein gene, so specific for the RSIV's major capsule protein. So I think that this is RSIV strain. So then, but I'm not sure about the genotype 1 or 2. So I tried the whole genome sequence. Then I tried the BLAST search or some analysis for the nucleotide identity. That strain is included to the RSIV's grade 1 or grade 2. So it means maybe different from the ISKNV and TRBI. Thank you very much.
Any more questions? If there are no more questions, thank you again for the excellent presentation.
Thank you very much.
Now we'll move to fourth presentation by Omkar Vijay Bragdi. Yeah. So characterization and investigation the dietary effect of co-administration of Shionella putrescens and rhodovolum sulfidophyllum on gut microbiome, changes in forefinger, thread fin, tetradactylum species.
Okay, good evening everyone. I'm Ashish from India, but I'm now doing my PhD in Taiwan, in NPUST. So my topic of presentation is characterization and investigation of the dietary effects of co-administration of Shionella putrescens and rhodovolum sulfidophyllum on gut microbiome changes in forefinger thread fin, Eleutheronyma tetradactylum. Sorry. So I'd like to begin with this presentation with the aquaculture and its problems. So we have witnessed the growth of aquaculture in the past few decades, which has invited the problems such as diseases from the pathogens such as bacteria, fungus, parasites, and virus, which has led to the economical losses of the farmers. So the farmers are trying to use some of the methods to control this disease. The traditional control methods are use of antibiotics, vaccines, and chemical treatments. But this has some of the problems like antibiotic resistance, cost of vaccines are very high, and chemicals might leave the residues. And now in the recent commercial aquaculture, people are moving towards the sustainable options such as probiotics. This is now considered as a viable alternative because it is having no such problems as traditional methods. And when it comes from the same host, which is autochthonous probiotics, they have the minimum adverse side effects. So in this study, we are focusing on two strains. One is Shionella putrescens. From here on, we will be referring it as S5, and rhodovolum sulfidophyllum. The first one, the Shionella, is first isolated from the Senegalese sole, which then demonstrated the inhibitory activity against the Photobacterium damseli subspecies Piscicida. And also, the studies have found that it has reduced the susceptibility against some microbial infections. It has shown the effective against like listed pathogens such as Legionella anguillarum and Vibrio harvei, whereas rhodovolum sulfidophyllum is a non-sulfur bacteria which is widely used in marine aquaculture as probiotics. In the fish, it is demonstrated to be upregulation of the several immune genes, and also it promotes the growth of beneficial bacterial populations in the gut. The candidate species of this study will be forefinger threadfin. This fish was chosen because it's one of the important food species of in Taiwan. Especially its growth is concentrated in the southern part. People choose this fish because it is excellent food resource. It grows fast and it has a remarkable meat quality. So the method will be identification and characterization of the strains that we have mentioned. So in the identification, the preliminary identification will be from the staining, that is gram staining, and then it will be molecularly sequenced using 16S RNA. And characterization, there will be two-part, growth parameters and antimicrobial properties. In the growth parameters, we will be testing it against some of the antibiotics to check whether they have some antibiotic resistance or not. And we will be simulating the gut by various amounts of bile salt and pH. And also halotolerance, we will be simulating the environment because this is a marine probiotics. And in the antimicrobial properties, we are going to take cell-free supernatant and also the dialyzed and concentrated cell-free supernatant to test its inhibitory properties. For the in vivo evaluation, the probiotics will be first lyophilized, the strains will be lyophilized and coated onto the pellets, and then it will be checked for its viability. And then in the experimental design, there will be 4 treatments. One is S5 alone and FA120 alone, and combination of both and control, whereas it is uncoated feed. So this feed will be fed at 4% of the body weight daily, and the sampling will be done at 8th day, taking 3 number of fishes, and then the intestines will be sampled to send the sequencing for the 16S RNA. And then the results, the strain identification, we found out that both of these probiotics were gram-negative. Only difference was S5 was a short rod and the FA120 was the long rod, and they both showed hemolysis negative when we tested again in the blood agar. And they were identified using 16S RNA gene, and then the sequence was blasted against NCBI and to confirm the strain. For the antibiotic susceptibility profiles, our strains were susceptible to most of the antibiotics that we tested, which is one of the key parameter, because it might have the risk of horizontal gene transfer of the resistant gene. And then the gastroenteric and environmental stress tolerance revealed that FA120 was superior compared to the FA S5, because in the pH and in the bile, we can see the growth is better in case of FA, but S5 has a dip. And whereas in the salt concentration, various different concentrations of salt, S5 and FA almost was the same. And then the antimicrobial properties was tested, like as I said, cell-free supernatant and also dialyzed cell-free supernatant. So we saw, for the first, we just selected the cell-free supernatant, where we checked S5 and FA both. So S5 was having some effect against Photobacterium damselae and Streptococcus ineae. And in the second stage, we just dialyzed and concentrated the cell-free supernatant to concentrate the proteinaceous compounds responsible for this inhibition, where we found better results, as we can see the halozones over the discs. And for the viability of probiotics, we checked after we coated onto the feed. We tested by suspending the feed into the PBS, and then the bacteria was grown in the plates, and then it was sequenced to check the strains are really the probiotic strains that we are interested in. And the metagenomics analysis revealed that the combined feeding showed the colonization of Shivonella at up to 12% in the short term of 8 days only. When controlled to and the one interesting thing that we want to see here is in the control group, we have the domination of Vibrio vulnificus in the upper, the majority of the control group was occupied by the Vibrio vulnificus, whereas in the treatment, it got reduced. And in all the taxonomic levels, the result was same. Shivonella was able to it was able to colonize in the gut. And for this, the logic, what we suggest is the probiotics might have combinedly had the synergistic effect, because we also did the monostrain treatments from S5 and FA120 alone, but it did not show any, or it was poorly colonized when they were done in the monostrain, but they had when they were co-applied, the colonization was evident. So what are the possible mechanisms are, we are trying to, we are proposing these tripartite mechanisms. So one might be a pathogen suppression. So FA120 has in the studies have witnessed to have antioxidant enzymes from the host. It can induce the antioxidant enzymes, and also it has sulfur metabolites which might have disrupted the Vibrio quorum sensing. Niche modification, where FA120's photoheterotropic nature altered the luminal redox potential, creating the microaerophilic conditions which might have facilitated the S5's growth. And metabolic cross-feeding, FA120 is known to produce the extracellular polymeric substances which might have provided the carbon source for the S5, and S5's sideropores countered the Vibrio vulnificus iron piracy potential. So in the future work, we are going to extend this work. This was a pilot study that we have done, so we will be extending it for the longer term to evaluate the gut microbial dynamics. And in the next, we are going to study what exactly the functional antimicrobial components that are produced by S5, because they are having the inhibition zone. And also, we are going to challenge the probiotic-fed fish by some of the selected piscine pathogens to see that they are really protecting in vivo. And final, I would like to acknowledge DAA for providing me this stage to explain my studies, and my university, my department, NSTC, Taiwan for funding our project, and Taiwan ICDF for funding my PhD in Taiwan. Thank you.
So anyone questions? If not, I have one question. Whether after establishing the colony that you got, whether refeeding is required or not? After 8 days, you have shown that is the establishment of colony in the intestine. After that, whether refeeding is required or they can continue?
Sir, as of now, in the short term, we are able to see that it has reduced the colonization of the pathogenic strains. But in the long term, we weren't able to answer this, like if the refeeding is required or the long-term effect, how does long-term effect is better than this or it is worse than this? So until then, we are not able to tell this, sir.
So then coating, every day you are giving coating?
Yes.
So then how you are making the culture, then the culture you are...
No, we coat the lyophilized feed and we are going to store it at 4 degrees until we feed.
So after lyophilized, that lyophilized culture?
Yes.
So how you are giving a coating to the feed?
We are using the vegetable oil for coatings.
Pardon me?
Vegetable oil.
Okay, but you know how much that, freeze-dried sample will be there?
Like the cell number is 10 power 8 or 10 power 9 per gram, sir.
So it is visible, is it feasible to practically hand coating is possible?
Yes, because the bacteria are growing in plates. As of now, like it's like a proof of concept. So I don't know about the feasibility in the commercial.
Okay. Thank you.
Thank you.
Thank you, sir.
So our fifth presentation by Dr. Brandon Rafael D. Jesus Castillo, Korea. Yeah, he will be presenting on WSSB proteins drive glutamine metabolism through glutamate dehydrogenase activities in Lithopenias vannami. Your time starts.
Okay. Good night, everyone. My name is Rafael. I'm original from Nicaragua, Central America, so very far from here. Representing Taiwan, I'm going to present about my research that I'm doing my PhD, also about the WSSB proteins, how these drive one specific metabolism, exactly the glutamate, pointing out one enzyme that is the glutamate dehydrogenase activation in the shrimps. So how do we know white spot disease is still one of the most deadly viral in the shrimp culture? Most about these clinical signs that we can see about this red body coloration and this white spot in the exoskeleton, with a cumulative mortality that it can go for the 2 to 10 days for completely 100%. The main cause for this virus is the white spot syndrome virus. In this figure, we can see a brief about the virus replication. It more enter the host cell for endocytosis process. And there's more cellular and molecular processes happening there in order to get the virus genome replication. This is a double-strand DNA virus, a very big one, and had more than 500 putative open reading frames, where mostly the 90% of this one is like remain unknown. He had from previous evidence from molecular and cellular studies, he had proved that this WSSB is able to induce, for example, the Warburg effect, the glycolysis, nucleotides, and also lipogenesis. But here we make some emphasis about the glutamate metabolism because it's more used as an alternative source of carbon and nitrogen for replications. Before also the glutaminolysis model was proposed that happening in this during WSSP. So there is 3 specific enzymes, for example, the GDAs, ACHT, and alpha-ketoglutarate. So research find out that this activity is being enhanced by the virus. But this information make us some questions like how really this is happening. So we were trying to hypothesize that there is some protein interaction that is happening to activate these GDAs. And And as we know, one protein is not alone. Mostly it's a multi-complex protein happening there. And we would like to find some new insights about WSB and also host protein binding during the regulations. So for this, we're using different models to prove the protein interaction. The first one is the yeast to hybrid screening. So we use the EH109 yeast strain, and we created an AD library. So this AD library have different open reading frames, and our bait is this host M-site GDH. So we just put it together, and we need the AD and the BD interaction to start these different reporter genes that will help the yeast to grow. After this, I got 5 candidates, 5 WSB open reading frames that are able to interact with the GDH. So these are my five purity candidates for further experiment. After this, we go to the cell line. This is the SS9 cell, that insect line, because we still don't have a shrimp line. So firstly, I went to do some transfection and express these proteins. So among these 5 WSB proteins, I was able only to express these 2, the 079 and the 164. Then I use the same system, but I use a co-immune precipitation. This is to prove the interaction between these 2 proteins. So in here, we use 1 protein as a flag, and then we use some flag beads, and this has to pull down the B5 protein. So you can see, we can see the pull down for the GDH. And at the same moment, I prove the 079 and the Waxin4. And this also show us that, they can interact with the GDH, but however, they also are able to interact each other. That might form a complex when they are expressing. Then I will go to the in vivo model. So I designed a double-strand RNA to silence these 2 genes. This first graph shows us that the silence efficiency was successful. Then I went to check the GDX expression during the 12 hours first, and then to the 24 hours. And then we used 3 main point genes, that the AN1 WSB, the BP28, some structural gene, and the ICP11, and also we checked the genome copy number. So this data gave us some insights that these genes are very crucial for the WSB replication. Now for the second part, we use an AP-MAS strategy that we express a recombinant protein, then we incubate with some infected gill lysates with co-immunoprecipitate, and then we send it to the LP-MAS to form some proteins correlation network. At the meantime, we need to do some data filtering, and we got a proteomics database, so we adjusted to 2x differential protein expression, and then we try to map or associate WSB proteins, and we can create our correlation network. So for our first network, we can see like the red color, this means as a positive correlation, the green one as a negative. We identify some pathway related for the DNA of hemocyanin. But the interesting is we find some hot proteins like the pCNA that proliferate nuclear antigens, some of them unknown, some of them related to actin-based cell adhesion and DNA replication. That's for the first network. The second one, it got a little more complex, but we also see the pCNA, and then some differential protein in between, and we got also the CHMP4, that is the shared multivesicular body proteins. Also, we find some signally scaffold and some signally receptor also involved in this correlation network. So try to summarize all this together. What we're presenting here, this is a strategy to investigate virus, more exactly to the pathogen and host correlation network. And we were able to find 2 proteins that interact and activate this GDA's enzyme. But however, our correlation gives us new insight, more of that related to the viral entry for endocytosis process, and some of them for the viral budding. So our correlation network at the end is giving us more questions instead of answer us. The further research might be needed to try to explain how these immediately early protein or early protein are really happening here to be a multifunctional modulator factor proteins in all these replications. So I would like to acknowledge for my advisor, Professor Han-Xin Wang, also the guidance for the Professor Luo, Sisuen Lao Sư, Professor Tu, and our T-Lab group here. Thank you very much.
Thank you. Thank you for the excellent presentation. If there are one or two pressing questions, I would request. So if there are no questions, again, thank you for the excellent work.
Thank you.
Then our next presentation will be the sixth presentation, last but one. So please have another 10-20 minutes patience. So this is by Su-Wen Chang. Glutamate pyruvate transaminase is a key metabolic regulator during WSSB infection in Letopinis-Benami. Please.
Hello everyone, my name is Shu-Wen Zheng. I'm glad to attend the DAA12 and share my story. And I come from Taiwan, NCKU. Let's just jump into the topic. About white spot disease, its clinical signs include calcified exoskeleton and the reddish discoloration on the whole body and appendages. And because it can spread horizontally and vertically, also during the epidemic, the mortality rate can reach 100% just within 3 to 8 days. But until now, there's no effective treatment or prevention methods. And the pathogen causing this disease is white spot syndrome virus. The intact virion have an oval shape of it, and it contains 3 layers, contain the lipid-containing envelope, tegument, and the nuclear capsid. And its genome is a large single-cellular double-stranded DNA molecule. Size is from 300 to 307 kilobase pair. And today I'm major focusing on WSSV-induced metabolic reprogramming. As previous study already understand the virus infection will induce host metabolism reprogramming to benefit virus replication, include energy production, viral genome replication, envelope or capsid formation. So go back to WSSV. After WSSV infection, our previous result already shows cell will prefer to use oxidative glycolysis instead of aerobic respiration. And we also observe a Warburg effect like metabolic reprogramming. The hallmark of the Warburg effect is the high consumption of the glucose, but also show the accumulation of the lactate. And we also observe other WSSV-induced host metabolism reprogramming, include nucleotide biosynthesis, lipogenesis, and glutameolysis. So put this information together. After WSSV infection, we found the glucose in consumption is increased, and most of the carbon source flux into the lactate, and that caused the intermediate of the TCA cycle. Acetyl-CoA is insufficient. So cell to overcome this situation, they will use the glutamine as another carbon source to conduct the glutameolysis and go into a reductive glutameolysis. The protein I am major focusing on is involved in this pathway. And it can catalyze a reversible reaction between glutamate, pyruvate, and the alpha-ketone glutarate and alanine. And why we're focusing on this enzyme is because different metabolites involved in a lot of important pathways. The first is pyruvate. Pyruvate can use for the lactate formation, and this can help to regenerate NAD+ and allow glycolysis continue go into the lactate production. And also, it can help in the high oxidative stress. It can directly remove the H2O2 and produce the acetate, which is another carbon source for the lipid synthesis. And glutamate, it can be turned into alpha-ketone glutarate by a lot of enzymes, including glutamate dehydrogenase, aspartate transaminase, and the GPT, and which can provide cell to use the glutamate in a more flexible way. And alanine, which can connect the amino acid metabolism and the glucose metabolism. About the alpha ketoglutarate, which can directly replenish into the TCA cycle, and which is a central precursor for the amino acid biosynthesis. So first step, we are going to look into the shrimp GPT-1. In our in-house database find out, we only have GPT-1 this isoform, but actually in a human, we have GPT-1 and 2, and they are located in the, one is mitochondria, another is cytosol. So we would like to figure out, in the shrimp, this protein is majorly expressed in which side of the cell. So we express the recombinant protein in the SF9 cell. Then we can see the immunofluorescence result. The green ones mean the recombinant GPT-1 protein, and the red signal is mitochondria. After we merge these two images, we can find the GPT-1 is expressed majorly in the mitochondria. Then we wanted to know, after WSSV infection, whether it will affect the GPT-1 gene expression or protein activity. So we collect the sample after 12 and 24 hours post-WSSV infection. The result shows that at 12 hours post-WSSV infection, no matter mRNA or protein activity, both are significant increase. But protein activity is reduced after 24 hours post-infection. Then to understand the importance of GPT-1 during WSSV infection, we use a double-stranded RNA to knock down the GPT-1 mRNA expression. And wait a minute, okay. And after we knock down the GPT-1 mRNA gene expression, we found it significantly reduced the WSSV gene expression and the genome copy number. It is mean this protein is quite important for WSSV replication. And next, we would like to know this signaling pathway already been proved involved in WSSV-induced Warburg effect. So we would like to know whether this gene also been affected by this signaling pathway. We use specific inhibitor to inhibit the protein in this signaling pathway. And the result is a little bit complex. Let's directly conclude the result is that RAS shows us an inhibition modulator for GPT-1, but mTOR-C2 is an activator. Then, because we would like to know how GPT-1 contributes to WSSV infection, so first we silence GPT-1, then replenish the metabolite, include glutamine and alpha-ketoglutarate. So first, we confirm the silence experiment is successful, and after we replenish this metabolite, first we focus on the after we replenish alpha-ketoglutarate, actually they didn't rescue virus gene expression. And if we replenish the glutamine, only ICP11 shows upregulation. But let's look into the genome copy number. Both of them didn't rescue the WSSV replication. So far, we only can observe the glutamine slightly increase ICP11, but WSSV genome copy not rescue after any metabolite replenish. So go to conclusion and the future work. First of all, we knock down the GPT gene expression and observe WSSV genome copy number and gene expression. Both of them was being significant downregulate. And RAS is inhibition modulator for GPT-1, and mTOR-C2 is an activator. And after we knock down the GPT, then replenish with glutamine and alpha-KG could not rescue the WSSV replication. So in the future, we will continue try to replenish another metabolite to see whether is pyruvate or alanine involved in the WSSV infection and help them to to produce a lipid or to involve in amino acid metabolism. And in the end, I would like to thank DAA12 gave me a student travel award and have this chance to share this research. Thank you so much.
Thank you. If there are any questions, maybe one question. Half the audience is gone. So if there are no questions, thank you so much for the excellent work and the excellent presentation. Thank you. So our last presenter, Athia Azzahida. She will be presenting on dietary perilla leaf extract supplementation enhances shrimp immunity and disease resistance.
Good afternoon, everyone. Firstly, I would like to thank to the DAA that provide me the Student Travel Award. So today, I'd like to share my research that kind of different from my previous lab mates. So today, my research entitled Dietary Perilla Leaf Extract Supplementation, Enhanced Shrimp Immunity and Disease Resistance. And this study actually explore novel and natural approaches for improving shrimp health. So talking about the global shrimp production, I think many speakers has been sharing a same thought that shrimp farming has experienced remarkable growth over the past few decades. And countries like China, India, Thailand, Vietnam, and Indonesia are leading in the shrimp production. But the rapid intensification of shrimp farming has brought a serious challenge, including the suboptimal growth rate, often triggered by the environmental stress fluctuating from the salinity and temperature to the nitrogen, which creating an ideal environment for pathogen to promote the disease outbreak like happen. So with the challenge, improving the shrimp health and disease resistance has become a top priority in aquaculture. And one promising strategy is the use of the bio-based functional shrimp feed additive, which offer multiple advantages, including the bacterial pathogen inhibition, and then enhance the shrimp immune system, also improve the digestibility. And one of the promising candidates among the many plant-based options is the perilla. Why perilla? Because this plant is widely used for the traditional medicine and well known in human health. And for the distribution, this perilla, or mostly commonly named as a Chinese basil, mostly found in the East Asian country, and also can be found in India and Thailand, so make it more accessible. And related to the bioactive compound, perilla contains several functional compounds, including alkaloid, terpenoid, flavonoid, and peptide that has been demonstrated to provide the antioxidant, anti-inflammatory, and also antimicrobial effect. Interestingly, some study in fish has demonstrated that perilla extract has been found to enhance the immune system of aquatic organisms, so leading to the improved disease resistance and reduce the stress level. So, but it raises a question that could this perilla also hold the potential as a functional feed additive in shrimp? And that's what exactly I'm work with. So here are the phytochemical characterization of perilla leaf extract, or PLE. So based on the proximate analysis, it contains a high level of the nitrogen-free extract and crude fiber, which important component for gut health and metabolic activity. And focusing on the HPLC, focusing on the bioactive compound, our HPLC profile confirms the presence of the rosmarinic acid, which is a flavonoid that has a strong antioxidant capacity, as shown in this DPPH assay. So the rosmarinic acid shows the DPPH percentage reaching near 90% at just 0.5%. Millimolar concentration indicates that it has a strong ability to neutralize free radical even at a lower concentration. So to evaluate the potential of this perilla leaf extract in a shrimp, we conducted a feeding trial and then continue with appen immersion challenge, and using the several challenge group, including TSP S02 as a control for non-appen causing Vibrio parahemolyticus and 5-HP as appen causing Vibrio parahemolyticus. So what happened when the shrimp are fed with the perilla? So most significantly, after appen challenge, the 1% shrimp feed show a higher survival and a clear protection compared to the non-supplemented group. And visually, the difference was also clear that the shrimp in 5-HP no PLE display pale and damaged hepatopancreas, while those in a 1% PLE show healthier and brown hepatopancreas. And then at the molecular level, both of the pure toxin and plasmid copy number also significantly reduced in a shrimp feed with a 1% PLE compared to the no PLE. And this suppression was confirmed at the protein level using the ELISA. So then to further evaluate the immune function, we tested the antimicrobial activity from hemolymph against appen causing Vibrio parahemolyticus. So interestingly, after challenge with both of the S02 and 5-HP, the 1% PLE feed group show a stronger bacterial inhibition compared to the non-PLE group. And this effect was not seen in the TSB and no bacterial challenge, suggesting that PLE supplementation might enhance the antimicrobial properties from the hemolymph only during the bacterial infection, not under normal condition. So then we focus on the phenol oxidase activating system, which is important in the shrimp defense. And at 40 days post-feeding, we observe upregulation of the some of the key immune genes in the PO system, including the SERPIN7 and ProPO. And after appen challenge, we also observe another spike that the 1% PLE sustained a stronger upregulation of the genes. So what does this mean? That PLE not only activating the immune system in early function, but also sustain a stronger immune response during infection, and potentially explaining the reduced pathogen burden and also disease resilience we saw earlier. So we also observe the upregulation of the superoxide dismutase, indicating the enhanced antioxidant capacity, and also lysozyme, which play a role in the bacterial clearance. Interestingly, for the ROS modulatory gene expression was significantly lower in 1% PLE compared to the no PLE. So this suggests that PLE might prevent the excessive ROS signaling, maintaining the immune balance, and also protecting a shrimp from the oxidative damage. So to confirm that gene level finding, we also evaluate the enzymatic activity, including the PO activity that show that the 1% PLE has a higher PO activity and elevated the superoxide activity. And consistent with the ROS modulatory gene expression, the ROS production showing lower in 1% PLE. So this enzymatic assay support the hypothesis that PLE dietary supplementation activate the PO pathway and antioxidant response, supporting the shrimp defense against appen infection. So then, to validate our finding about PLE's protective effect, we evaluate the hepatopancreas tissue integrity using H&E staining. So as you see here, under normal condition, the hepatopancreas has well-organized tubule lined with the intact epithelial cell, but then after appen challenge, as we observe in the no PLE, show the typical appen lesion, including the slugging of epithelial cell indicated with the black arrow, and also the massive hemocytic infiltration and melanization. In contrast, 1% PLE still maintain the intact hepatopancreas structure, indicating much less tissue damage observed in the PLE group. In addition, a quantitative scoring of hepatopancreatic features has confirmed this observation. So as you see here, in 1% PLE was significantly a lower score for the necrosis and immune cell infiltration. Meanwhile, for the lipid storage as an indicator for immune function and also the energy reserve was highest in 1% PLE. So all this reinforced the hypothesis that PLE could protect the stream and reduce the oxidative damage. So to conclude, our study demonstrated that dietary supplementation of perilla leaf extract enhanced disease resilience in shrimp through a multiphase mechanism. First, it can reduce the appen virulence by lowering the toxin purine B and plasmid copy number. The second, it can enhance the hemolymph antimicrobial activity, particularly under active bacterial infection, and then trigger the immune response by upregulating of the key immune gene. And then now I'm still working on the microbiome metagenomic analysis to evaluate whether PLE can promote the abundance of the beneficial bacteria and also modulate the specifically functional pathway related to the immune genes. And I would like to acknowledge the AFS Society that provide the student award and my advisor, Professor Han-Ching Wang, and also Professor in NCKU for helping me and the T-Lab member. And thank you very much.
Any questions from the audience? Yeah.
Yeah, thank you for your nice presentation. I just want to know, have you identified any active ingredients for this antibacterial activity? Active ingredients, have you identified any?
Yeah, actually from the perilla leaf extract contains several bioactive peptides that potentially act as an antimicrobial. But for now, we're still working on the figure out whether that can also help to inhibit some of the bacteria other than the Vibrio parahemolyticus.
Another thing is that tannin. Have you estimated the tannin content of this perilla leaf? Because tannin is the inhibitor. Tannin. Tannin.
Yes, tannin as a part of the component of the perilla. No, because from our HPLC analysis, it's already show the signature of the perilla leaf extract is a rosmarinic acid. So we focus on that functional bioactive compound that act as an antioxidant capacity.
Okay, thank you. Please check the tannin content also. If tannin is present, that feed may not be that much effective. If tannin content is very high.
Now that depends on what type.
No, we focus on the rosmarinic acid.
Okay, thank you.
Thank you.
Now that depends. What type of extract it was, this side? What type of extract? Is it aqueous extract, ethanolic extract, or what type of extract?
For that, actually we receive a raw material of the perilla from a company. It's formed like a powder. And then because we don't represent that company, so we could not share that. But actually this perilla is widely produced in each country, and we can get easily the powder of that perilla. So that's our concern why we developed this.
Okay, thank you very much.
Any more questions?
Yes.
If there are no more questions, thank you so much.
Thank
Wonderful
you very
work.
much.
So with this, we come to the end. We had 7 very, very wonderful presentations from 7 very young minds. So anyway, thank you all. It was excellent listening to all of you.
Nothing to say more because all are waiting for the dinner. So because these are the travel award presentation, they haven't already been selected, and the selected candidates only they have presented. So thanks to all the participants for this presentation at the last moment, extended dinner hours. Thank you very much.